All right. Well, thanks for coming this morning. My name is Mike Behnke. I'm an assistant professor at the School of Veterinary Medicine here in Baton Rouge, LSU, and the Pathobiological Sciences Department. I just wanted to say for Shreveport and New Orleans, please interrupt me if you have any questions. If I don't make something clear, if you have questions about the slides or anything, please pipe in during the talk. It would be informal, almost like a lab meeting type talk. So I'll just get right into it. My lab studies the parasite Toxoplasma gondii. So today I'll be talking about the biology of this parasite. And this will be the general outline of the talk. Kind of go over the different portions of the life cycle and the biology of the parasite, then get into some of the recent studies we've done to look at the genomics of the parasite, to use genetic mapping to understand phenotypic differences between parasites and to identify virulence factors. And then a project we're currently working on in the lab, the main focus of the lab is to understand more about the enteric stages of the parasite. And these are the stages of the parasite that develop within the gut of a cat, the definitive host. So Toxoplasma gondii is an Apicomplexan parasite. Apicomplexan designation is a phylum of parasites. They're all single-cell protozoan parasites, intracellular, and they follow a general, I'll use the mouse for the pointer so this shows up on remotely. They all generally use the same type of life cycle. So there's a proliferative stage where the parasites expand in number, and then they go through, this isn't working very well, they go through the definitive stages or the sexual stages. And this is usually in the gut, this is in the gut of some sort of animal. So for instance, in Toxoplasma, for Toxoplasma, it's the gut of cats. In Plasmodium, it's the midgut of a mosquito. And so this is where, that mouse isn't really working, so I'm going to just use this pointer. The proliferative stage goes into the gametocytes, and these are the sexual stages. So they form male and female gametocytes, which fuse, form a zygote. And then this is usually some, in the case of coccidium parasites, an environmentally resistant form that contains sporozoites that can then differentiate into the proliferative stages again. So this is kind of a pared down life cycle that all these parasites share. So they all share this definitive stage, male and female form. Again, toxo in a cat, Plasmodium, midgut of a mosquito. Amirias, very speciated genus, and each species of Amiria goes through the gut of a different animal. So there are Amirias that only infect poultry. There are Amirias that only infect goats or sheep. And then Cryptosporidium, which you probably heard of, Cryptosporidium parvum and hominis, that parvum can infect cows and humans, and then hominis can infect humans. Now, the one major difference between these parasites is some are kind of single host parasites, so they only have the definitive stages. They're monoxenous, or they have an intermediate stage portion to the life cycle, which includes Toxoplasma and Plasmodium. The monoxenous are Cryptosporidium and Amiria. So Cryptosporidium and Amiria only go through the gut of their definitive hosts, producing those sexual stages and the oocysts, and they're shed into the environment, and then they have to go back through the gut to continue through this proliferative stage. Toxoplasma and Plasmodium are different, where they have their definitive host stages, in the cat and the midgut respectively, but for instance, Toxoplasma can infect every warm-blooded nucleated cell, warm-blooded animal nucleated cell. So that's mammals and birds. And so it can infect mice and humans in these intermediate stages. And these are proliferative stages that also contain a dormant stage, but which are respectively called tachyzoites and bradiozoites, that can be passed from intermediate host to intermediate host through the ingestion of uncooked meat. And the classic part of the life cycle for Toxoplasma is an infected mouse with dormant bradiozoites in the brain or something gets eaten by a cat, and then those bradiozoites can go through the gut of the cat, complete the life cycle into the sexual stages. And so all these apicomplexans kind of have this pared down life cycle, and they also share, as part of their name, this apical complex, which is important for invasion. So a lot of these parasites have this conoid, which is important for attachment and invasion, and then various organelles, either called Rhoptries or micronemes, that secrete proteins upon attachment and invasion, and either help with the active invasion process or help to set up the vacuole that the parasite resides in as it pulls itself intracellularly and sequesters itself away from the host cytoplasm. And a lot of these proteins traffic back to the parasitophorous vacuole and help protect the vacuole from the host. So this is kind of outlined a little bit better here in a cartoon. We've got these big bulbs here called Rhoptries, and then these smaller microneme dots. They're both secreted upon attachment and invasion. So for instance, this is Toxoplasma attaching to a host cell. It reorients itself on the apical end, and as it does that, it secretes micronemes and Rhoptries and forms this moving junction. It's a ring of proteins, both complex between parasite and host proteins, that the parasite can pull itself into the host cell through and then form this parasitophorous vacuole. So it's essentially sequestered away from the host cytoplasm, which would kill the parasite, and sets up a nice home. So a lot of these proteins then either are important for this moving junction and other aspects of the active invasion process, or traffic to the vacuole and help to give integrity to that vacuole. And so you can see this here where we've labeled intracellular parasites with a couple antibodies, either to ROP1 or ROP18, and either doubly stained here, red and green, yellow, with DAPI is the nuclear staining of the parasite. So you can see those two Rhoptries kind of ringing the parasitophorous vacuole. Some of these other proteins traffic to the host nucleus and actually modulate the host gene expression after invasion. So make a nicer home for the parasite to reside in. Another unique aspect to the apicomplexans is they have 3 genomes. So they got the nuclear genome. Toxoplasma has 14 chromosomes, about 65 megabases, about 8,500, 9,000 genes. Plasmodium's a little bit less in terms of gene number. It's about 5,500 to 6,000. So it's got a standard nuclear genome, and then like any eukaryote, it's got a mitochondria. But the apicomplexan, a lot of them, most of them, have an apicoplast. So this was, like the mitochondria, sequestered through an endosymbiotic event. But interestingly, it was a 2-stage endosymbiotic event. So there's a primary endosymbiosis with the ancestor algae that took blue-green algae, prokaryote, to make the karyotic algae. And then that algae was endosymbiosed by the ancestral apicoplast, or apicomplexan parasite, to form the apicoplast. And so interestingly, the apicoplast has 4 membranes, which is suggestive of this 2-stage endosymbiotic event. Now, originally, this algal cell that became an organelle of the apicomplexan parasites had a lot of genes, but over time, those genes moved to the nucleus. So the apicoplast genome is very small, just like the mitochondrial genome, 35 kb. But a lot of those genes have moved to the nucleus, so now they're made in the nucleus, and the proteins traffic back to the apicoplast. So this has been an active area of study in the field, both for Toxoplasma and Plasmodium, because they have apicoplast. And this organelle is derived from plants. So if you can know what the apicoplast does and study the genes and target drugs to plant-like proteins, you're going to have better chance of designing drugs that might target the parasite and not hit a mammalian gene. So this has been an active area of study. Found that the Apicomplexan is important for fatty acid biosynthesis and also isoprenoid biosynthesis. The unique thing about Toxoplasma is, of course, it's got that definitive host life cycle that goes through the cat, but it has the intermediate stages, and Toxoplasma can infect pretty much every warm-blooded animal in the world. So people have gone out and done seroprevalence studies in all sorts of animal populations, from whales to mice, different birds, and they found a certain percentage of those animals to have seropositive titers to the parasite. This is in contrast to other parasites like Neospora caninum and Haemondia. That Haemondia goes through the gut of a cat, completes its life cycle, but it seems to only infect rodents and doesn't cause infection and disease in all these other animals. Neospora caninum is similar to Toxo, except that it goes through the gut of a dog to complete its life cycle and really only affects ungulates as the intermediate host and doesn't seem to affect humans and a large variety of warm-blooded animals. So Toxoplasma seems to break out of this intermediate life cycle restriction to infect all these warm-blooded animals. And so it's very prevalent around the world. And this translates, because Toxoplasma can infect humans, into quite widespread prevalence around the world in humans. This prevalence is at different rates. So here in North America, about 10 to 20% of the people have been exposed to Toxo. Again, this is looking at blood, and 10 to 20% are seropositive, have antibodies to the parasite. In France, historically, it's been higher. So 40 to 60% of the population has been exposed to the parasite. And in Brazil, it's even one of the highest areas. Particular areas of Brazil, it's up to 80% of people have been exposed. One interesting study recently came out in Germany where they did a very large study, thousands of people, and found about 50% of the population in Germany has antibodies to the parasite. And the last sentence in the paper suggested that it's known that you can get the parasite through uncooked meat, and the Germans love their pork sausage. And so if you don't cook it properly or cure it, you can transmit the parasite. So it's likely their predilection for pork. In France, it's famous for eating tartare, raw meat of various sorts, and so that might be the reason why it's a higher prevalence rate. Here in the States, there was a push in the 80s to make sure the pork meat is cooked at 165 degrees, and so the prevalence rates have been lower here in the States. Over time in France, it seems like with education, people becoming aware of this, prevalence rates have gone down over time. Now, in most cases, people become infected and they won't even know it, or they might come down with what they think is the flu. You get a interferon gamma response, might be sick for a couple days, and then if you have a competent immune system, your immune system is going to check the parasite, the taxozoite growth, and the parasite, either it can wipe it out or the parasite can differentiate into this dormant phase called the bradyzoite. So it's thought that people who have been exposed to the parasite, have antibodies to the parasite, may be lifelong carriers of the parasite because of this dormant cyst. So if your immune system crashes, the parasite can recrudesce from that dormant phase back into the taxozoite, which is a rapidly growing form, and cause disease. And it can do this in the brain of humans. So during the HIV/AIDS epidemic, in the beginning, in the 80s, a lot of people were dying from toxoplasmosis because there's this background prevalence rate, immune systems were compromised, and people, of course, were dying from secondary complications, and a lot of those were toxoplasmosis. Another disease caused by toxoplasma is congenital. So if a woman hasn't been exposed to the parasite and becomes infected during pregnancy, she doesn't have antibodies to it, so it takes a little bit longer to check the parasite growth, and the parasite is sent into the fetus. and it can cause syphilitic type diseases in children. So 200 to 400 children in the United States are born with some sort of form of toxoplasmosis in the United States every year. Here we say the recommendation is that women, if they have cats in the home, they shouldn't handle the cat feces, have the husband or someone else in the family deal with the cat, or adopt the cat out, and after you have the child, bring it back into the house, because it's the feces where you can really get infected from that oocyst form. In France, they actually test women throughout their pregnancy for seroconversion. So they find out they're pregnant, they test them to see if they're positive. If they're positive, they're okay, because they have antibodies, and they don't need to worry about being reinfected. But if they're negative, then they test the woman throughout pregnancy. If she converts, then they can put her on anti-toxoplasma drugs to prevent larger rates of congenital toxoplasmosis. And then also another immune privileged area of the body is the eye. So we can get toxoplasma cysts forming in the retina, so you get these ocular lesions. And so anytime you go to the eye doctor, they're going to be looking for this in your eye. But again, people with normal immune systems can control the parasite and really don't suffer from these effects. Now, you may have heard of Toxoplasma in the news the past couple of years. If you recognize this guy as Martin Shkreli, he's actually, I think, in prison right now for another scheme that he had. But at one point, he bought a company that made the only drug approved for Toxoplasma in the United States called Daraprim. And at the time, they were selling Daraprim for, I think, $1 a pill. And he jacked it up to $13.50 a pill. Before that, a couple years before that, it was $1 a pill. The company bought it, jacked it up to 13. Then he bought that company and jacked it up to $750 overnight. So he cornered the market, because here's a drug that there's only one drug for Toxoplasma. So this became a huge news item for a few weeks, kind of a poster child for drug companies jacking up drug prices. The problem with this story is that Daraprim is pyrimethamine. And this drug has been used since the 1950s for Toxoplasma. So it's a generic drug. Any company can make it. There's no patent that gives him exclusive rights to this. So it was a generic drug where he jacked it up $750 overnight. And what you didn't hear was that Express Scripts, about 6 months later, was going to offer Daraprim for $1 a pill, because they could make it as generic. But this got a lot of news a couple years ago. It's the only approved Toxoplasma drug, pyrimethamine, and not many people use it. About 1,000 people in the United States every year use it. So another area where you may have heard Toxoplasma in the news is its ability to affect the behavioral changes of infected hosts. So does anyone know who this guy is? This is Ted Nugent, and he had an album called Cat Scratch Fever. So this is what cat scat fever. Thing is, this guy might be the head of the NRA here in the next few years, so I'm sure he'll do a good job. I'm sure you'll hear about him in the news. So these behavioral changes come from, or studies come from mice, where researchers would put either rabbit urine or bobcat urine in an area, a caged area, and let the mice just roam around this area and monitor where they would hang out in the area. And it was found that when there was bobcat urine and the mice were uninfected, they stayed as far away as they could from that urine for most of the time. But when they were infected with Toxoplasma, they tended to actually get nearer to the urine, same bobcat urine here. So this led to these behavioral ideas that mice lose their fear of cats as the surrogate bobcat urine when they're infected with toxo. So here is a parasite actively changing the behavior of this intermediate host And it's an intermediate host that would get preyed upon by cats, and so that the parasite could get back into the cat and go through the definitive stages and the sexual stages where really get recombination and complete the life cycle. So these are interesting studies. It depends on what strain of mouse you use and what strain of parasite you use. It's about 50-50. People see this or they don't. There are also rat studies that show this. A lot of ideas of how this is happening. But it doesn't seem to be the parasite is actively doing it. There's a study where we've got a parasite that can induce this behavioral change, but gets completely wiped out by the immune system. So there's no dormant phase hanging around in the brain that might be causing these long-term behavioral changes. And the behavioral changes persist. So it may be the immune response is causing some of these changes, inflammation. And these are ideas that are being pursued in terms of behavioral changes and immunity. Because you see behavioral changes in other situations and other infections, influenza, these types of things. These mouse studies are specific to Toxoplasma. So this led other people to look at these possible behavioral changes in humans. And so you do association studies with humans. Of course, you can't actively infect them, but you look at a particular population, see what the prevalence rate is, and for those that are positive, are they exhibiting behavioral differences compared to those that are uninfected? So here's the mouse studies. And so some of the human studies have seen that increased traffic accidents for Romanian truck drivers that were toxopositive, so they're a little more reckless in terms of their driving if you've been exposed to the parasite. Less altruistic is another study where it saw an association. Again, these are small effects. And then one that's been known for a while is this association with schizophrenia and seropositivity. Again, these effect sizes are small, and these schizophrenia associations have been seen with other infections. So sometimes, occasionally, you'll hear about these stories in the news with Toxoplasma, either the Daraprim story or these behavioral stories. But I like this quote from Robert Sapolsky, who's actually worked in this area. And he's written some popular books on his research looking at hormones and stress in animals in Africa. And so he was interested in this in terms of a parasite being able to modulate the behavioral of animals. And so he says these effects are incredibly cool, but the effect sizes are really small. And so if we want to reduce traffic accidents, let's really focus on using our smartphones while doing that. So just a grain of salt there, these behavioral effects, their associations are small effect size. So to get back into the life cycle more completely, this is specific to Toxoplasma's life cycle. So we'll start here with a cat ingesting a chronically infected mouse that has these bradiozoite cysts in the brain. Once these bradiozoites get liberated from the cyst as it travels through the stomach into the small intestine, these bradiozoites can infect the enterocytes of the small intestine and turn into the first of the enteric stages called the merozoite. So this is a proliferative stage. You get many different numbers of merozoites growing in the small intestine, and then those merozoites can differentiate into the male and female forms, the micro and macro gametes, that eventually fuse to form an oocyst. And so they go through a round of mitosis and meiosis and get shed into the environment as this environmentally resistant oocyst that's unsporulated. And then over the course of a few days, it sporulates into two sporocysts that each contain 4 sporozoites. So these are the single cell actively invading forms. There's 8 of them in that oocyst. So if this is ingested by an intermediate host or even a definitive host, then these sporozoites can infect that animal and then turn into the actively, or differentiate into the actively growing form called the tachyzoid. The tachyzoid is the cause of disease in intermediate hosts. So if it's growing in the brain, causes plaques, encephalitic type diseases. If you mount a proper immune response, then you can stress the parasite out, and it will go into this bradiozoite form, possibly for the life of the individual. And then if you get preyed upon by a cat, those bradiozoites can, again, complete the life cycle. So it's not really known what the major route of infection in humans is, but it's likely uncooked meat. So different animals have different tropisms. The parasite has different tropisms for creating bradiozoites in different animals. So in beef, the meat is, there's not many cysts in beef. Although a cow can become infected, but a lot of cysts in the brain. So parts of the cow that we don't eat. But in pork, the meat is cyst burden is high. So that's why you'll hear cook your pork meat for these types of parasites. But you can become infected directly from the oocyst, and the sporozoites can eventually, so either the oocyst sporozoite or the bradiozoite. So you can eat an infected animal, and those bradiozoites can regress back into a tachy. So here are a few pictures of some of these forms. This is all in the cat. So the merozoites, they have these banana-shaped forms, so they look a lot like the tachyzoite and the sporozoite. And again, these are the proliferative stages, so you get high numbers of parasites of these. And then they differentiate into these microgametes, which are flagellated forms of the parasites, so two flagella here. Kind of cool to see swimming around. And then macrogametes, so it's the female form. So this microgame is going to fuse with the macrogamete, produce these oocysts. So this is an unsporulated oocyst, kind of a undifferentiated mass here that eventually sporulates into these two oocysts. The cool thing about these oocysts is they autofluoresce under UV light. So, and that's one cool way to see them under the microscope. And then here we've done an IFA on an infected cat gut. You can see how the infection kind of rings the tip of the villus. All these villi are infected with parasite in red and yellow here. So it's just that one layer thick along the intestine of the enterocytes where the parasite differentiates into these definitive stages. Any other cell, it's going to be a tachy or a brady. So here are the tachyes. Again, you got a banana-shaped form. This is a rapidly growing form, and they divide kind of by binary fissions called endodiogeny, where one parasite goes into two and then two into four. And once they divide and get a huge vacuole, they can lyse and then reinvade cells, and you can get these plaques. And then a bradiozoite cyst can have hundreds to thousands of parasites within it, but they're really dormant and not growing. So this cyst is kind of sequestered away intracellularly from the immune system, can hide there for months to years, and maybe occasionally checking the immune system to see if it's okay to go back into the tachyzoite form and grow, but if not, it'll stay as a brady and hope to get into either another intermediate host or a cat. So these are pretty much dormant, arrested in their cell cycle. These guys, they divide every 6 to 8 hours, so they're really rapidly growing. So if you've got these growing in your body, it can do a lot of cell damage. And again, the sources of Toxoplasma infection are mainly pork. So cook your pork meat. We'll get into some of the studies we've done recently on this parasite to understand more. First, about the genomics. We were interested to understand what the population structure of the parasite is worldwide because it's so prevalent, and what those genotypes may tell us about its biology. So initially, we started as a community isolating the parasite from Europe and North America, because that's where a lot of these studies were done in the 80s and 90s. And it was found that the parasite genetic makeup was fairly clonal. They were very genetically restricted into 3 types, types 1, 2, and 3. So that was kind of an interesting find. One note is that cat remains have been found with humans associated with human burial at least 10,000 years ago. So it may be that this spread of clonal types was due to domestication events of cats, and that these types were conducive to going through domesticated cats. Although we just don't really know what led to this population structure. Then over time, more isolates came in from around the world, and particularly South America, and we found that the genetic diversity is much higher there in South America. And this is expressed here in this slide, kind of busy, but what I want to point out is for Europe, North America, and China, Africa, it's a lot of blue and green with a little red. So these are types 1, 2, and 3 parasites. So mainly prevalent there, whereas in South America, you get this rainbow of genotype. And so much higher genetic diversity coming from South America. So this really shows that the parasite likely evolved in South America and at some time spread from there, but we don't know when. Was this something that happened with the Columbian Exchange? Or was it much longer ago, and some of those, a few types were able to spread throughout Europe and North America. And so, one idea here is that there are a lot of cats in South America, and there are also some pretty big rodents, like nutria. We've got nutria here in Louisiana that were imported from South America, pretty big rodents, and then there's also capybara. And then you've got jaguars and ocelots and other cats here. So maybe that's the ancestral life cycle for the parasite in these wild felids and rodents. And so looking at genotyping, this represents over 1,000 parasites. Collapsing those genotypes into groups finds that there's several clades with about 16 haplogroups. And different ways to look at this. This is done with RFLP with just a few genetic markers, about 11 to 15. And this is all genomic markers that was done after a genome sequencing project. And essentially, the parasites collapse into the same networking. And so they're basically 6, 7 different major groups called clades for these parasites. So out of this, we sequenced 62 genomes that represent the most unique aspects of the genetic diversity around the world for these parasites, coming from these different haplogroups and clades. And looking at the genome sequence, there are different aspects to that we can find. And so one is that we found large segmental duplications and aneuploidy in some of the strains. And so this is where one strain has an extra copy of chromosome 1A, and another strain has an extra copy of chromosome 3. And then other chromosomes and other strains have smaller duplications called segmental duplications. And so this may result in a phenotypic difference in these parasites, some sort of selective advantage. We don't know, but it's one characteristic from genomic data that you can look at. So along with this, you get large duplication events. You can also get smaller duplication events called copy number variation, where particular genes are duplicated. So you've heard of selection and different ways that genes can change and be selected for. So you've got your DN-DS ratio in terms of mutation rates in a gene that can be a signature of positive selection. So that gene is changing more rapidly than the background rate of all the genes in the genome. So there may be selection pressure on that gene to find new function. Another way that that selection pressure can happen is by having copy number variation. So the gene can be duplicated. One copy can retain its original function, and the other copy can mutate and possibly find new additional functions. So this is one way to look for genes that are under positive selection that may be giving selective advantage to the parasite. So this is a Circos plot of all the different chromosomes, 14 different chromosomes of the parasite. And these are all the genes along the outside. And then along the inside tracks are different gene families that show, with the larger circle here, some evidence of copy number variation. So these genes have either tandemly duplicated, so they copied in at the location, or there's some other evidence of copy number variation. Some of these, you've got 15 copies of one gene. Some of them, there's only another copy, just 2. The interesting thing about these, a lot of these genes are Apicomplexan specific. So there are Rhoptries and micronemes that I talked about before that are important for invasion, Gra proteins. So surface antigens, and then this new family called TG-FAM that have all been expanded in greater quantity than the rest of the genes in the genome. And so an example here, you can see how this might be viewed from the genome data itself, is where we plot the gene copy number as you move across a chromosome. And this is chromosome 12. So for, I think this is 10 kb sections, you can determine what the duplication rate is. And so for most genes, it's right around 1. But a few, it's a little higher. And so, for example, here we've got this section that contains this Rhoptry protein, Rhop5, that's got about 10 or 11 copies higher than the background rate of the chromosome. So doing a genome study, one thing you would want to look at are genes under positive selection. And so that's ENDS. Those are non-synonymous mutations in the gene. And then also copy number variation. And so this is one way to represent it, Surcos plot. If you're into genomic data, you'll get used to some of these Surcos plots and how to read them. So this Rhop5 is, we'll talk about a little bit later. Another aspect we looked at in the genome was relatedness. So another way to look at how the parasites collapse phylogenetically. And what we found was that if we compare 2 strains to each other, we can look at the SNP rates and found that certain strains have regions that are completely shared between strains. So as an example here, we've got strain ME49, and we compare the genome to itself. There are no SNPs, right? Because it's the same strain. There are not going to be any differences at the genomic level. So that's just a representation of that, 0. If we take another parasite that's very similar, ME49 versus ARI, you can see there are regions that they completely share. There's hardly any SNPs, so like chromosome 1A here, and various other regions on the chromosome as you go across 14, and then other regions where you got higher SNP rates. So then ME49 and CAS, they share chromosome 1A completely, but the SNP rate is much higher across all the other chromosomes. And then ME49 against Coog, they're pretty much different. So what this basically suggests is that there's admixture going on between parasite strains. So we compared all 62 to 62 comparisons and determined what the % similarity was between these comparisons and collapsed this into a hierarchical plot. And basically, they collapse into the clade structure that was determined by a different method here. So essentially, this suggests that the genetic structure of Toxoplasma is being driven by recent admixture. So this means that 2 parasites are to be found at the same time in the gut of a cat, and they can go through sexual recombination and then produce progeny that are a combination of those 2 parents. That's admixture. And so that's shown here with ME49 and ARI. So they are share parental genotypes where the SNP rate is very low. So this showed to us that there's a lot more sexual recombination going on out in the wild than we had previously appreciated. So I'll get into genetic mapping, and this actually translates directly from this admixture idea into how can we use this part of the biology to understand more about the parasite. So one of the big questions in the field was, what are the genetic factors that account for the virulence differences in the parasite? So what we, over time, using these original types that were isolated from Europe and North America, found were the types 1 were virulent mice, so you could inject 1 to 10 parasites in a mouse, and that mouse will be dead in 7 to 10 days. Type 2 were moderate, depending on the parasite load and the mouse background, and type 3 were essentially avirulent. You can inject 1,000 parasites into any mouse, and they won't even get sick. So what is the genetic difference that's causing these virulence changes? What factors account for this? So we can use this idea of using a genetic cross or sexual recombination in a cat to do QTL analysis, quantitative trait loci mapping. So what we do is we feed 2 parental strains where we know the phenotype. So types 1s are virulent, type 3 are avirulent. We co-feed those to a cat. They each have a different drug selection. They'll go through sexual recombination, and then we can clone those out and genotype them either by double drug selection or just cloning, and find progeny that are a combination of the 2 parents. They're recombinant. And we only are interested in progeny that are unique. We don't want to clone the same parasite out and use that. So all the progeny are informative in that they're unique and that they are a combination of type 1 and 3. And so we can look at that using, this is an older method. We're now just sequencing the progeny and determining what the genetic makeup is. But back in the day, we used this RFLP mapping where we would PCR up a section of the genome and cut it, and you can see banding differences that are reflective of either type 1 or type 3, and you can call that in the progeny. So that's one thing you need to determine in the progeny is the genetic makeup of all the progeny across the genome. And then the second thing you need to determine is the phenotype of those progeny. Are the progeny more like the type 1 parent or the type 3 parent, virulent or avirulent? So you can combine those two where you have a genetic map of each progeny across the genome and its phenotype, whether it's like virulent or avirulent, and do an association study across all the chromosomes at each marker that you have for genetic information and determine, is there an association between the phenotype and the genetic marker makeup? And if there is, you get these peaks, and you get a high LOD score. So these are QTL peaks. And so in the case of a 1x3 cross, we saw 1 peak on chromosome 7A, and this was narrowed down to about 20 genes. And so you don't get directly to the gene. You get a locus based off the recombination events that you get in your progeny. But you can look at other data to try to find out what gene in that locus might be the important one. And one piece of data were gene expression data of the parents, types 1 and 3. And it was found that all the genes at that locus were expressed similarly in both types 1 and 3, except Rhop18, which was not expressed in type 3. So this is, again, one of those Rhoptries I talked about that's secreted upon invasion. And it's actually an active kinase that's secreted inside the host cell and is critical for virulence. So if you express a Rhop18 in a type 3 parasite, it turns it into a virulent parasite, just that one gene. And so then we also did this with a 1x2 crossed them, looked at the progeny and the phenotype, mapped this to chromosome 12, got a big locus here, and found that there was one gene here with the copy number variation different. And this was Rhop5. So Rhop5 had 11 copies in type 2 and only about 5 or 6 in type 1. And really, it's the allelic structure of these genes that result in the phenotypic differences. So if we knock out Rhop5 from type 1, it renders it avirulent. So now a parasite where it would take one parasite to kill a mouse, now you can inject 1 million parasites and the mice don't even get sick just by knocking out this locus. So what's going on here? We've got Rhop5 and Rhop18 that we found are virulence factors using this genetic mapping method. And what other studies have shown is that both are secreted upon invasion and traffic back to the parasitopher's vacuole. Rhop5 actually recognizes these host immune-related GTPases. These are host proteins that are upregulated upon interferon gamma stimulation. So once the host realizes it's been infected, it mounts an immune response. You get interferon production and a host of genes that are expressed. Some of these are IRGs. What they do is they try to traffic to non-host vacuoles and kill them. Rhop5 seems to bind to these IRGs and then allow Rhop18 to phosphorylate it, preventing it from going to the parasitophorous vacuole. So if you have both of these virulent forms, an active kinase and the virulent locus of Rhop5, they can phosphorylate these IRGs and prevent the innate immune system of the mouse from destroying the vacuole. And so the parasite continues to grow, lyse, and reinvade, and then eventually kills them out. And so there's some IFAs here. I'm kind of running out of time, so I'm going to skip ahead. Another aspect of this is you've got expansion of the Rhop5s, is again another indicator of positive selection. And the interesting thing is these IRGs have been expanded in mice. So up to 20 different copies, more than 20 copies, expanded in mice or different IRGs. So maybe this is partially an arms race where Rhop5s are trying to create new copies to be able to interact with the IRGs, and the IRGs are trying to escape those interactions. And IRGs are important for other intracellular pathogens, Listeria. One caveat is humans don't express IRGs. We've got two. One's kind of a pseudogene, and the other is only expressed in the testes. So it doesn't seem that we have IRGs that are functional in terms of an immune function. There are other genes similar to IRGs, though, that humans do have called GBPs, and these are actively being studied in terms of their ability to limit intracellular parasites. So I'm going to leave it there, kind of right at 50 minutes. Was going to talk about a project we're working on right now where we're trying to understand more about the enteric stages of the parasite by studying these forms in tissue culture, using new methods for actually growing. We can grow little mini guts in tissue culture, so we can isolate intestine, grow these out in organoids, and this is what Mississippi is doing, and grow these mini guts for the first time. This was figured out about 10 years ago. So we're adapting this to various veterinary animals that are definitive hosts for these parasites. Historically, it's been impossible to really grow intestinal tissue cell for long periods of time in tissue culture. Now this is possible because of being able to keep the stem cell happy that's in the crypt of the intestine. And so these continue to grow. And so what we're hoping to do here, you can see an IFA, what they look like. Kind of got these crypt-like regions. So what we hope to do is to make monolayers out of these epithelial cells from cat intestine, and then infect these with bradiozoites of the parasite, see if we can induce the enteric forms of the parasite in tissue. And if we can do that, then we can open up a whole new area of study for the enteric stages of these parasites, which are right now intractable because you have to use the animal to do that. So I'll leave it there, and please, if you have any questions, let me know.
So just to wrap up, can you take 5 minutes to tell us how did you get to where you are today? Because it never seems to be a straight line for anyone, and everyone has a unique story. So if you could take 5 minutes. I don't know if you did that at the beginning.
No, I didn't.
Okay.
Yeah, I didn't talk about. So I went to undergrad at NAU, Northern Arizona University in Flagstaff, studied microbiology, did the standard 4 years, and then after that, did not go into grad school right away, so kind of took an unconventional route. Went to work for various computer companies. So I learned how to script and code command line techniques there, mainly Unix, so that was really helpful. So I did that for about 7 years, and then was kind of getting sick of the corporate lifestyle and wanted to get back into science. So found an ad for someone to do some bioinformatics type work, or who knew Unix, computer skills, to work in a parasite lab. So I took that, got the job, and I worked in the lab for about a year before deciding to go to grad school. And so that was a Toxoplasma lab, and so that was at Montana State University. And did 6 years of grad school, standard route. And then, so there I studied the cell cycle of the parasite. So we did microarray studies where we could study hour by hour. We could synchronize the parasites in terms of their cell cycle and look at how the parasites progress through the cell cycle for the first time. Also was looking at the differentiation of the parasite from the tachyoid brazoid stage. Just one thing I forgot about, as an undergrad, I worked, what really got me into kind of working in a lab was working in a soils lab. There's a forestry department there at Flagstaff, northern Arizona. So that was a lot of fun because I got to go, half the time I was out in the field collecting samples, water samples, soil samples, and then half the time I was actually looking at mycorrhizae on the roots of trees in burned and unburned areas. So that was kind of fun. Started out washing dishes, of course, so you get your foot in the door. So this summer is good for you guys because you're actually going to get great laboratory experience. And that exposed me to what life is like in a lab. And then so years later, after grad school, was fortunate to get a good postdoc in St. Louis, WashU, in David Sibley's lab, and he also was looking at Toxoplasma. There, wanted to do a genetic cross, so did the 1x2 cross looking for virulence factors, and that's where we found Rhoptries. And then also started some of these enteric stage studies with the parasite. And so that kind of study is something completely different than anyone's really doing in the field right now. And part of the reason why I got the job here was able to get funding. And so took a job here 3 years ago at the LSU School of Veterinary Medicine, working on toxo in this area that really hasn't been studied too much, at least.
So it sounds like as an undergrad, you had no idea you would go into research like this, right?
No. Yeah. I knew I liked science and microbiology, but yeah, there's no way to know where
you
you're
got swept
be.
up with the computer revolution. And,
yeah.
And then got
Computers
tired
really was the fortuitous thing for me. Gave me kind of a unique aspect, is knowing Unix and being able to deal with some of the bioinformatics issues.
Especially now, you can take advantage of that experience for the high performance computing.
Yeah.
If you need it for your data analysis and stuff.
Yeah. So kind of an unconventional route, but you can always get back into it. Get into a lab or take a conventional route.
Well, thank you so much. So
yeah. Any questions?
let's start with questions. I always start with remote sites. So Shreveport, I'm going to go north. You guys have any questions? You guys are going to have to unmute, I believe. Shake your heads no, or raise your hand that you're going to talk. I guess not. Ryan's walking up. Oh, that's Ryan. He's the operator.
All right, go ahead. All right, so I think I missed the part, but is there any kind of test to test this kind of parasite on the laboratory? Like for the human beings, what are the tests? I think I missed that part. And are there any kind of vaccination developed till now for this parasite?
Go ahead and repeat that,
Yeah,
because you're not coming through clearly enough.
So I'm just asking about the tests, if there are any kind of tests for that in the lab, for us, and is there any kind of vaccination developed till now for this kind of parasite?
Testing for the parasite and vaccinating for the parasite? Is that what you just said?
Yeah. Is that what you're talking about? How do you test to see if you're positive?
Yes.
Yeah, there are various tests. One is ELISA-based type test. It's testing to see if you have antibody to the parasite in your blood. And there's various other commercial tests people use. either trying to stain for live parasites in the blood, but it's mainly a seropositivity test, antibody-based test.
Can you get rid of them? I think was the other part.
Right, is there a vaccine? So there are probably hundreds to thousands of papers where people have expressed recombinant proteins and shown some sort of vaccination in mice. Would you want to do that for a parasite that largely doesn't cause a problem in most of the population? You know, I talked about those prevalence rates. I didn't mention that based off of those, it's estimated maybe up to a third of the human population has been exposed. So would you try to vaccinate everyone for a parasite that's so prevalent? It's really not causing disease in most cases. It's only when you have immunocompromised situations or unique situations such as congenital, which there are procedures in place to try to prevent that. So I don't think a vaccine, even if it were possible, because we don't have a vaccine for any type of parasite, i.e. malaria. I mean, there's one that's proved its effectiveness can be argued. But even if there were a really good vaccine, I don't know if it would essentially be used other than in very unique situations, but not across the whole population. But a good question. Yeah.
Yeah. New Orleans. We're short on time, so I'm gonna just. New Orleans. You got any questions?
And again, the drugs are very effective. Pyrimethamine. So if you know that you're positive and you think you need the drugs, you can go on the sulfadiazine pyrimethamine regimen, and it'll clear up the parasites real good. At least the actively growing form. It's the chronic form that you cannot clear. Yes.
New Orleans?
Can you hear me okay?
Oh, yeah, loud and clear.
Okay. I just have one question. It was about the stem cells and like growing the like intestinal thing. Do you mind going to that slide?
I was hoping might have a little bit more time to explain it. So real briefly, because I was running out of time. So historically, it's been you can't grow intestinal tissue in culture. You might get a primary cell line to grow a couple times, but then they'll die out. So being able to study these cell types has been intractable until about almost 10 years ago now. Sato and Clevers figured out how to grow intestinal cells by using a conditioned media. So they found that essentially R-spondin and noggin are required, and then also Wnt3a, which can be added to the media. These factors, they're proteins that induce signal events that either induce the stem cell to grow or prevent deactivator. So the noggin is actually preventing an inhibitor. So if you make a conditioned media with these three factors, then you can isolate people have done this for small and large intestine and stomach. You can isolate these little ileum, and you can isolate the crypts, which at the base of the crypt is this LGR5 region, or this is where the stem cells reside. And so here's a blow up of that. You've got these LGR5 stem cells, and basically that's what's being induced to continue to proliferate in tissue culture. And so what we use is a 3D matrix matrigel format, and these tissues can grow into what are called organoids, or specifically for the intestine, enteroids, and they can grow in 3 dimensions, and you can get these nice round structures. Sometimes they pinch off and make their little crypts. And so this was done for mouse and human, and so when I came here, I wanted to possibly use this new technology to study intestinal cells from animals that serve as definitive hosts for these types of parasites, specifically cats. But we also did this for various other animals that are definitive hosts. And we're able to grow these cells out using the standard conditioned media that's used for mouse cells and human. And so now that we can grow these out, here's an IFA showing proliferation at crypt-like regions. So they're mini guts, enteroids. We found that cat cells require nicotinamide uniquely. That kind of threw a loop. but mainly it's so now we want to use these cells to give the parasite the one cell type it needs to differentiate into these enteric stages. So we can finally grow those cells. Now we're adding the parasite to see if it's sufficient to induce them to go into the enteric stage. Because any other nucleated cell, the bradiozoite is going to go into tachyzoite. And only in the cat epithelial cell lining of the small intestine will it go into the merozoite.
So New Orleans, did that cover it?
Yeah, it did. Thank you.
Yeah, thank you.
So I'm going to switch to local audience. Y'all have any questions here? All right. Well, thank you so
Okay,
much
thank
for
you.
speaking to our students. And we're going to take a quick 5-minute break.